And then in 1952, French scientist Chargaff published the rules, so to speak, of base pairing. This was an important finding. And that was, again, 1952. Again, I'm writing these dates just so you know them. I'm not going to ask you dates. But you should know the names of these scientists and what they did. In his work here, Chargaff's work, there were some scientists, Rosalind Franklin, Francis Crick, and James Watson, who discovered the structure of DNA, or were able to determine the structure of DNA as being a double helix. And that was in 1953, when that Nature paper was published. There's a lot more to the story, as I suspect many of you know, between Rosalind Franklin and the data she generated, and how Crick and Watson used that data without her permission. We could certainly talk about that at some time, but not really relevant to this discussion right now. But it's an important part of history. During this time of understanding inheritance and discovering the properties of DNA, the idea of epigenetics, that is, the idea that somehow or another, the genes that were being discovered and talked about in these papers, there had to be some way of controlling that. So the idea of genetics was first used in 1930. However, it wasn't coined, the expression epigenetics, until 1942. So in 1942, when it was coined by this scientist named Conrad Waddington. He was a well-known geneticist and developmental biologist. He thought of epigenetics, when he first coined the term, as this causal mechanism by which genes of the genotype bring about a phenotypic effect. So he recognized that these genes then made some kind of a product. Didn't know it was a protein at that time, but some kind of product that gave that phenotype. At the time he made this discovery, they didn't really understand what a gene was, so he wouldn't have been able to add to how these genes were being controlled, but that would come later. So today's definition of epigenetics, as I see in your textbook here, which is stated nicely, I think, a study of changes in gene function that are meiotically or mitotically heritable. And importantly, they do not change the DNA sequence. But Waddington was the first to kind of begin to use this term in a way that was getting after the effects of the genes. During this early time, geneticists they would have thought heredity, evolution, development, and traits in general caused by the genetic material. However, people who weren't geneticists but were biologists, I mean geneticists are biologists too, but another group of scientists felt that these things up here, heredity, evolution, development, and traits, were caused by cytoplasmic factors. There was a scientist by the name of Nanny. And then he was more of the biologist type, where he felt that the cytoplasmic factors were important in controlling these events, but he did feel that these cytoplasmic factors were important, but the genetic system also factored in. So he might have been the first to kind of start to bridge this gap. Another scientist, Boris Efruzi, he classified genetics as the transmission of genetic material. But epigenetics as the functional state of the nucleus. So he was a very strong proponent of epigenetics. And he felt that even at this early time, and this was late 1950s, where he was making his comments and his studies. He often reminded colleagues that not everything that is inherited is genetics. Meaning there was something else involved too. What it was, he didn't know. At the same time as Boris Efruzi, another scientist, Lederberg, and I'm just going to mention his name briefly here, but he was really important in bringing this idea that not everything that is inherited is genetics. He felt strongly that there were things outside of the gene that were also important when it came to inheritance, transmission of traits, expression of traits, evolution. And so the work of these two scientists really started to lead to this idea of the acceptance of epigenetics as a field. Doesn't mean it was solid, didn't mean they understood everything that was involved with it, but they accepted it as a field. And there wasn't some formal announcement that today we accept epigenetics. It wasn't anything like that. It was just over time what happened is you stopped seeing definitions of epigenetics in papers. So if a paper was published and they wanted to talk about epigenetics in the early 50s and late 40s, they would have defined it or debated it or felt it had to be defended. But by the late 50s, early 60s and beyond, the term epigenetics would just appear in papers, scientific papers, without the need to define it. It was a term in a field that was now accepted. One last person I want to mention, I think, don't hold me to that, but Marquardt here in 1968. So he jumped up quite a bit. He was a cancer biologist. He said there's probably no gene linked to cancer, but rather cancer is caused by a misprogrammed epigenetics. Now, in one respect, he was wrong. Now that we know there are certainly genes linked to cancer, but he was right on this. We now know that epigenetics is a major, major player in cancer. And we'll talk about that throughout the semester a little bit more, maybe a lot more. For some reason, I'm not sure why, but the decades between 1970s and 1990s, early 1990s, very little was done in the work of epigenetics. There were some things, but not a lot, except for one person. And I said that last guy was the last person, but I was mistaken. Holliday. It's important to mention Holliday because he gets a lot of credit for his work with recombination, but he also did something with epigenetics. And he showed that epigenetics could be explained by methylation. And that was in 1979. And it turns out this is one of the biggest keys to epigenetics, and it was discovered in 1979. And this work is often thought of as the seminal work that led to a lot of research in the 1990s on epigenetics. I do want to mention a couple other dates and historical events I think are important. And this goes back a little. This is a little out of sync, and I apologize. I should have mentioned this person earlier. But Mary Lyon, she was the first to show X-linked mosaic expression. And we're going to spend quite a bit of time talking about this later on. And this X-linked mosaic expression was due to X chromosome inactivation, what later were referred to as bar bodies, these unexpressed X chromosomes. In 1983, don't worry about the person, but extending from Holliday's work, they were able to show that methylation prevented regulatory proteins from binding. So we knew a few years before that methylation could cause epigenetics, but now they show that methylation actually prevented the regulatory proteins from binding to genes. They also showed around this time that methylation patterns could be inherited. In 1993, the phenomenon of imprinting was being explored. And we'll talk about this quite a bit as well. IGF-2 is a major player in here, insulin growth factor, and how that is differentially inherited whether or not you're male or female. 1994, a stronger molecular explanation of how cancer was linked to epigenetics. Throughout the 90s, most of the 90s, 1994 to 1998, a lot of really good research was done to establish the roles of histone acetylases and histone deacetylases were done. And these play a major role in turning genes on or off based upon the attachment of the histones. And these enzymes, they were first studied in yeast, but then throughout the decade, it was shown to be well conserved throughout all eukaryotes. In 2003, a very big thing happened, and that was the human genome was sequenced, or completed. This was a very big event. However, it opened up so many more questions. We have the genome, but now we have to understand how it's regulated. And a lot of that next step here, from 2003 to today, we're going to spend talking about over the next several weeks. Though there was a lot of information I threw at you here regarding the history of epigenetics, I really want you to focus on the major findings and the scientists that were linked to those major findings. Okay, now I'd like to move on to a summary of this podcast. In this podcast, we talked about genome sizes. And I should have stressed when we talked about genome sizes, I'm not going to ask you questions on a quiz or a test. What is the size of a yeast genome or a human genome or an amoeba genome? But the take-home point from that was that there are varying sizes of genomes, and you can't make any prediction about the size of the genome based upon the organism you're looking at in general. But what you should know about the genome sizes is that there has to be a mechanism to use those genomes. So then we spend some time talking about those mechanisms. So how is the genome used? And we spent quite a bit of time talking about euchromatin versus heterochromatin. In that discussion, we talked about histones and how they were linked to heterochromatin and its inactivity. We then spent the rest of the podcast talking about the history of epigenetics. And again, I'll stress, I'm not interested that you know the dates. I would like you to know the names of the scientists and in general what they did and what they contributed to the field. I would focus mainly on the ones linked to epigenetics, meaning I would probably be more likely to ask you a question about Waddington versus Watson and Crick. Okay, that ends this podcast. And in the next podcast, we're going to talk about how the genome is marked for epigenetics, and then the types of RNA molecules that we'll see in a cell, and then highlight some of the importance of epigenetics. All right, I'll see you in class. Bye.